Hello ladies and gentlemen, my name is Jacob Watson and I work in Dr. Amy Gancarz's Psychopharmacology lab, and we are investigating the effects of adolescent sucrose exposure on oxycodone self-administration in adult rats. Before I jump into the project, I want to share with you some statistics about the opioid epidemic and how it is devastating the United States. According to the Centers for Disease Control and Prevention, over 190 million opioid prescriptions were dispensed to Americans in 2017 alone. Of the patients prescribed opioids, nearly 29% abused them, and approximately 5% of them turned to illicit opioids such as heroin or fentanyl. From 1999 to 2018, nearly 450,000 people died from overdose due to at least in part by opioids. In 2018, nearly 70% of all overdoses in the United States involved opioids. With all this being said, opioids are extremely addictive and have a high potential for abuse. Drugs such as opioids activate a specific pathway in the brain known as the mesolimbic dopamine circuitry. This circuitry originates in the ventral tegmental area and projects to the nucleus accumbens and the prefrontal cortex. It is known that drugs of abuse hijack this pathway, but what is unknown is how environmental triggers such as natural rewards might increase one's vulnerability to drug addiction. In Dr. Gancarz's lab, we are currently investigating how natural rewards such as sugar influence drug vulnerability. Sugar is a ubiquitous, naturally occurring substance that holds strong hedonic value and is widely advertised all over the globe. In the United States, children and adolescents obtain approximately 16% of their overall caloric intake from added sugars. The U.S. dietary guideline limits sugar intake to 5 to 15%. Given that children and adolescents consume sugar so chronically, we are interested in studying how this may increase their vulnerability to substance abuse in adulthood. Our research focuses on the adolescent period because it's a critical time of development, and it is also a time where adolescents face an increased risk for substance abuse. With the prefrontal cortex not being fully developed, adolescents tend to have poorer judgment and are not really able to understand the long-term consequences of experimenting with drugs. There is also a significant behavioral and neuronal changes taking place, including within the mesolimbic dopamine circuitry. We know that exposure to drugs during the adolescent period increases future vulnerability to drug addiction. However, what we do not know is the impact that natural rewards like sugar have on drug-related behavior in adulthood. Previous work in Dr. Gancarz's lab studied the effects of sucrose exposure in the adolescent period on self-administration of low-dose cocaine. Here on the graph to the right, you can see the number of infusions earned over a 2-hour session on the y-axis across the days of testing on the x-axis, with the rats exposed to sucrose pictured in blue and the water control rats pictured in gray. Rats pre-exposed to sucrose self-administered a significantly greater number of infusions of low-dose cocaine. A progressive ratio schedule of reinforcement was used to determine the breakpoint. This is considered a measure of motivation. We found that rats pre-exposed to sucrose demonstrated a higher breakpoint compared to water controls. We hypothesized that rats pre-exposed to sucrose will self-administer a greater amount of oxycodone compared to water controls. To test this hypothesis, we used 10 male rats postnatal day 28, 5 in the experimental group, and 5 in the control group. We exposed them to non-contingent access to a 10% sucrose solution or water. Rats were placed in operant conditioning chambers for 30-minute sessions each day on a variable time of 1-minute schedule of reinforcement. This means that regardless of the rat's behavior, a reward was delivered on average of every 1 minute. Exposure lasted 15 days, and after this phase, rats were placed in the colony room with food and water available ad libitum until adulthood. But before we can start testing on oxycodone, the rats had to undergo a surgery that implants a catheter into their jugular veins so they can receive infusions intravenously. The rats were allowed 3 to 5 days to recover before testing commenced, and catheters were flushed every day with heparin saline to maintain patency. Once recovered, oxycodone self-administration began. Test sessions were 3 hours long and took place for 7 consecutive days. A house light was used to illuminate the chamber, indicating availability of oxycodone. Responses to the active side resulted in the infusion of 0.15 milligrams per kilogram per infusion according to a fixed ratio 1 schedule of reinforcement. This means that there was a set number of responses necessary to gain access to the next reward. Each infusion of oxycodone was accompanied by the onset of a 5-second cue light, which indicated that the oxycodone was delivered. This was followed by a 10-second timeout period where the house light remained off, and during which time responses to the active side were recorded but not reinforced. Responses made to the inactive side resulted in no programmed consequence. Here on the graph, Here the graph shows the number of infusions on the y-axis across the days of testing on the x-axis. The sucrose pre-exposed rats are labeled in blue, and the water control rats are labeled in gray. Preliminary results show that all rats exhibited a significant increase in the number of infusions self-administered across the days of testing. This indicates that rats had acquired the self-administration behavior. Furthermore, there was a non-significant trend that rats pre-exposed to sucrose self-administered more oxycodone across the days of testing. In the graph on the left, you will see the number of active responses emitted on the y-axis across the days of testing on the x-axis. Preliminary data shows a trend that rats pre-exposed to sucrose emit more active responses across the days of testing. These data show a similar pattern to that which was observed for oxycodone infusions. In the graph on the right, you can see the number of inactive responses on the y-axis across the days of testing on the x-axis. There was no significant difference between either group in the number of inactive responses emitted. These data are important because they indicate that rats are allocating their behavior to the response alternative, which produces the infusions of oxycodone. In this graph, it shows the relative frequency of active responses emitted on the y-axis across the days of testing on the x-axis. A value of 1 indicates rats are allocating all responses to the active alternative, whereas a value of 0 indicates all responses are allocated to the inactive side. These data indicate that rats pre-exposed to sucrose have significantly greater preference for the active side, which produces the infusions of oxycodone, compared to water controls. In conclusion, preliminary evidence suggests that exposure to sucrose during the adolescent period may increase vulnerability to oxycodone self-administration in adulthood. Rats exposed to sucrose during the adolescent period show significantly greater preference for oxycodone compared to water controls. There was a trend that rats pre-exposed to sucrose self-administer more infusions of oxycodone compared to water controls, and there was also a trend that rats pre-exposed to sucrose emitted a greater number of active responses compared to water controls. No differences were observed between adolescent exposure and inactive responding. Experiments are currently ongoing to increase sample size, and these data are not complete due to COVID-19 restricting lab testing. I would like to acknowledge my lab mates for helping me with this project, and my mentor, Dr. Amy Gancarz. I would also like to acknowledge the SRS program, the Mark U. Star program, and the Sally Casanova program. Thank you.